Right, hello everybody, good to see you all again, and we've got a fantastic 53 people already, and it's likely to be higher than that. So it's a great welcome here for Kersten Hall and the co-author of this wonderful book, The Dawn Fisherman, sounds mysterious, pretty plain, and the co-author Ralf Dahm, all the way from Germany. Ralph will be joining us later in the program, but right now we've got Kersten. You may remember Kersten from a previous talk about the crooked path, about incident. He's also written a wonderful book about uh, Professor Asprey and Leeds, who do all that work on war and DNA. Now, Kirsten, could you tell us a bit about yourself, and then we'll come back together again, and I'll give you a formal introduction.
Yeah, sure. Thanks, Peter. So my co-author Ralph and myself are very excited here to give you this double act this afternoon because the book will be out next April 2026, but I think you are the first audience to actually have the cover unveiled. So there's an accolade for you. My background is I was just explaining to Magda, I grew up in Leeds and I went away for 4 years as undergraduate, but I returned to Leeds for a PhD where I've stayed ever since. But my PhD was molecular biology, and I worked for many years as a molecular biologist before moving over into history of science. But I think there's a wonderful irony with that because as a molecular biologist, I spent most days in the lab cutting DNA, purifying DNA, analyzing DNA, working with DNA, but without having a clue who had actually discovered the stuff that I was working with. And I suspect I'm probably not alone in that amongst scientists. You work with this stuff, but you don't know who discovered it. Well, this afternoon, Ralph and I are hoping that in 45 minutes we can put that one right. So yeah, that's a little bit about me. I don't know if Ralph wants to come in on that and just give a brief introduction. If not, I'll hand back to Peter.
Okay, well, thank you very much, and thank you for allowing me back in to give you an introduction. Well, we're going to hear this wonderful talk, The Dawn Fisherman, Friedrich Miescher, and the Discovery in DNA by Kersten Hall, abetted by his German colleague, Ralf Dahm. And just before I say goodbye, until I come back to the questions, please put your questions in chat. I've always meant to say this verbally. I've put it in chat, but I'm not sure if all of you are aware of chat. It's right down at the bottom with two sort of beep bubbles. To click in that if you want to leave us a question, which I'm sure they'll be very interested to answer. So right over to you, Curtin, to give us the drum.
Oh, thank you, Peter. Thanks everyone for tuning in this afternoon. So here's the cover of the book that Ralph and I have been working on for the past couple of years. What I'm going to do is I'm just going to tell you a little bit about the story of this enigmatic person, the Dawn Fisherman, Friedrich Miescher, who he was, what he did, and why we think his story still matters today, and why it's important to remember him. And then I'm going to hand over to Ralph, who's going to tell you a really interesting story about a crucial part he played in remembering Miescher. So to kick off, I shall just start my phone so that I don't run over time. There we go. So you will be aware, I'm sure, that over the past week or so, there's been a renewed interest in the story of DNA following the death of James Watson. I think we can safely say that DNA now holds the status of being an icon in popular culture. You know, if you were to go out into the street and just ask anyone, I think, on the street about DNA, they would get that it does something really important. And I think, you know, that is thanks to cinema blockbusters involving Marvel superheroes and cloned dinosaurs, where DNA provides many a plot icon. Flick through the sports pages of a newspaper, you will see football managers talking about the DNA of their team. A few years ago, there was talk of the England team DNA. You'll hear politicians talking about it. I remember in the last run-up to the last UK general election, there was a studio debate between Rishi Sunak and Keir Starmer. Rishi Sunak warned his audience, don't vote Labour. They will put up your taxes. It's in their DNA. A few minutes later, Keir Starmer came back with, we will protect the NHS. It runs through our DNA. So, DNA, it's become iconic in the public consciousness. Ralph and I were looking around for examples of this when we were writing the introduction to the book. And one that I came across, you'll see it down on the bottom left, the trophy for the Grand National for the past 2 years has featured the famous double helix. But I think the best example of DNA becoming a cultural icon was the winner of the 2022 TV show Great British Bake Off, Dr. Shabira Yusuf. Her semi-final entry was a DNA double helix baked in Krokan pastry, which I thought was absolutely wonderful. And I got in touch with her, and she very kindly, both her and Love Productions, who do Great British Bake Off, very kindly have allowed us to use photographs of her her DNA pastry double helix in the book. So I think this, I'm willing to bet this is going to be the only book on DNA that will also include mention of the Great British Bake Off. So DNA is recognised as a cultural icon. However, if you then asked people who discovered it, then people are stuck. And as I said, I myself, even working as a scientist, even having done a biochemistry degree as undergraduate, worked for the best part of a decade without having known who actually discovered DNA. And I mean, the chances are, if you do ask people that, the names they are most likely to give you are Francis Crick and James Watson. But of course, they didn't discover DNA, they discovered its structure. Now, a good place to really start this talk is in the pub, specifically the Eagle pub in the centre of Cambridge. And I was in the Eagle pub a few years ago after I'd been doing some work in Cambridge. And I was sitting in the Eagle nursing a pint of beer, which actually went by the name of DNA. And I looked up and I saw this cluster of tourists all gathering around the wall opposite me with the hushed reverence of pilgrims visiting a medieval shrine. And it turned out they were clustering around this plaque that hangs on the wall of the Eagle, which says, inscribed in the plaque, it says, "Discovery of DNA. On this spot on the 28th of February 1953, Francis Crick and James Watson made the first public announcement of the discovery of DNA with the words, 'We have discovered the secret of life.'" And for one moment I toyed with my beer and I thought, would it really spoil their day if I was to tell them that the plaque is wrong? And it's wrong for two reasons. Firstly, there is that legend that Crick and Watson went bursting into the Eagle pub to tell everyone about their discovery. It didn't actually happen. But we might be able to discuss that at the end. More importantly, as I said, they didn't discover DNA. They discovered its structure, which was important, but the stuff itself had been discovered nearly a century earlier by Friedrich Miescher here. And as I sat and looked at that plaque, I thought, I wonder how Miescher would feel to see his discovery misattributed like that. After reflecting for quite some time on this, I think I've come to an answer. I think he would actually be delighted. By the end of the talk, I hope you'll appreciate why. But he's quite a tragic figure, I think, Miescher. He died crushed by a sense of really never having lived up to expectation, of opportunities missed. One of his former students famously described him as being like a ship that's sailing back to harbour laden with treasure, only to sink to the bottom of the sea just as it enters the port. So to introduce him, best place to introduce Miescher, I think, is on his wedding day. And you've got to picture the scene. There's his wife-to-be, Anna Maria Rush, and she's standing at the altar in the church, looking very nervous because her husband-to-be is nowhere to be seen. This is every bride's worst nightmare. And it turns out her husband is already married. Well, in a metaphorical sense at least, because a group of friends hurriedly form a search party. They go out to find the absent Miescher, and where do they find him? They find him at the bench in his laboratory, absolutely immersed in his scientific work, and they have to drag him from the lab bench to the church, reminding him he's actually supposed to be getting married that day. But I think that little scene, it gives you something of an insight into the guy and his dedication. I guess, I mean, perhaps alarm bells should have rung for his wife to be, because Miescher had this curious early morning habit of rising well before sunrise in the freezing cold winter months, and then making his way down through the freezing cold streets of Basel down to the banks of the River Rhine. The reason he was doing that is he wanted the Rhine salmon, or rather, he wanted their testes. Now, what on earth was the attraction of the testes of the Rhine salmon? Well, to understand that, let's take a step back. So, Miescher was actually born in Basel. It was his home city, and there were two key influences that shaped Miescher's career there. One was his uncle, distinguished professor of physiology Wilhelm His, and the other was a louse. And I've got to thank Professor Gareth Williams here, who wrote a wonderful book, Unravelling the Double Helix, in which he describes this louse as a louse that in its own modest way changed the course of scientific history. The reason for that is Miescher wanted to follow in the footsteps of his father, who was quite a distinguished doctor. But as a young man, he was bitten by a louse and he developed an infection which resulted in a hearing impediment. And he figured that his impaired hearing might mean his bedside manner as a doctor was not what it should be, so he began looking around for other career paths. And it was thanks to his uncle, Wilhelm His, who steered him in what he thought would be a particularly exciting direction. And that was the direction of a growing new scientific field that was called physiological chemistry, which I guess today we would recognize as biochemistry. And for that reason, His directed his nephew to the German city of Tübingen, which was an absolute crucible of this new field of physiological chemistry. Now, to understand why physiological chemistry was such a novel thing, to give you a bit of context, the main way of studying cells at that time was basically being done by cytologists, cell biologists, histologists, and their method was you take cells, you stain them with coloured chemical dyes, and you look at them down the microscope. Now, Miescher became critical of this approach. He thought the microscope approach was way too prone to artifacts. You know, you might end up observing what you think is a novel cellular structure, and it turns out just to be an artifact of the dyeing process. So Miescher was adamant we need a new way of studying the cell, and that new way was physiological chemistry, the approach of which was to characterize the cell in terms of its chemical components. And just what I want to plant in your mind right now is the thought that Miescher's criticism and his suspicion of the approach using microscopes, using cell biology, will turn out to be to be very ironic. So it was physiological chemistry that brought him to Tübingen, because Tübingen was the base of the man who would turn out to be Miescher's mentor, Professor Felix Hoppe-Seyler. He was one of the leading lights of this new field of physiological chemistry. Hoppe-Seyler had established biochemistry department in Tübingen Castle, and here you can see the state-of-the-art facilities in which Miescher was working. So this is his laboratory. So the task Hoppe-Seyler gave him was to work with white blood cells. Why white blood cells? Well, the problem at the time was solid tissues were not easy to prepare, whereas white blood cells are floating, free in solution. And I won't go into the full chemical procedure that Miescher used, but the raw material from which he obtained his white blood cells was, well, we're probably all thankful that we've all had our lunch by now, because a ripe source of raw materials was basically discarded surgical bandages that he picked up from the local hospital. Using those, he would wash the white blood cells out. He would wash the pus out of those bandages, And then he would begin the process of chemical analysis of the white blood cells. Now, what he was hoping to do was to characterize all the different protein components in the white blood cells. A little bit like, if you imagine, almost like a chemical zoologist. He's wanting to build up a chemical taxonomy of all the different proteins there. To his frustration, all he could really find was 5 different proteins, which had already been identified. What he did find, however, was a novel cellular substance that didn't chemically behave like a protein. Unlike proteins, it precipitated with weak acids to form this very cloudy precipitate. And Miescher made an interesting observation that the nuclei of these cells behave differently depending on whether they're in weak alkali or weak acid. In weak alkali, they swelled up, and he began to think that this novel substance he'd found was localized in the nuclei because of this connection with the behavior. So, because of its localization in the nuclei, he christened it nuclein. And to study it further, he needed to get way more nuclei. So he had to modify his preparation procedure a little bit, which involved another unsavory procedure that was washing the enzyme pepsin out of pigs' stomachs. And the advantage of pepsin was it gave him a really good way of separating cytoplasm from the nuclei. He was then able to obtain the nuclei as a fine white powder, and from that extract his nuclein. You can actually see, we think this test tube dates from the time he was doing this work, 1869. Today that's on display in the museum at the University of Tübingen. I've got to say, this was a first. He's the first person to separate cell nuclei from the cytoplasm. Now what he wants to do is he wants to do a chemical analysis on his nuclein. He does that using combustion analysis. And when he does that, what he finds is it contains lots of phosphorus, which you don't really find in proteins. So he's concluding what he's got here is a novel cellular substance that's not a protein. And I should say he's doing this. The conditions he's doing this are rather inhospitable. It's done in the freezing cold. And there's a bit he writes later on, he says, I cannot ignore the thought that it is here the essential physiological function of phosphorus in the organism is revealed. He goes on to say, an entire family of such phosphorus-containing substances will reveal itself. And this is a great line, so very laden, that this family of nuclein bodies will prove tantamount in importance to the proteins. Knowledge of the relationship between nucleus substances, proteins and their closest conversion products will gradually help to lift the veil which still utterly conceals the inner processes of cell growth. Very pregnant lines there, almost prescient in what they foresee. So what I'm going to do, I've taken my earphones out there. I hope people can still hear me. Let me know if they can't, Peter. Now, Miescher writes up this work. I should say, in actual fact, it's important to say the protocol he established here is pretty much, to a degree, basically the same. It's just what took him months and months and months to do. You know, a PhD student can now do in a morning using commercial kits. In fact, you can actually do it in your own kitchen using some strawberries, some washing up liquid, a bit of table salt and some ice cold vodka. You can actually do your own DNA extraction, which in principle kind of mirrors what Miescher was doing. So he wrote up his work because he wanted to announce this to the world. And his paper, however, wasn't published until 2 years after making the discovery. So he was doing the work in Tübingen in 1869. The paper wasn't published until 1871. When it was published, I don't know how good anyone's German is here, but it's titled Über die chemische Zusammensetzung der Eiterzellen. It translates as on the chemical composition of pus cells. It's not the kind of title that would have you sitting on the edge of your seat thinking, oh, I really must read that. It's kind of interesting he gives it that title. He doesn't say on the discovery of a novel cellular substance containing lots of phosphorus. Why the delay? Why the 2-year delay in this paper coming out? Well, the reason for that is his mentor Hopper Zeiler is cautious, and with some justification. Hopper Zeiler insists that he himself repeat all Miescher's work before it comes out. Why is he so cautious? Well, it's very much a case of once bitten, twice shy, because not that long earlier, another of Hopper Zeiler's students, a guy called Oskar Liebreich, he had also claimed to have found a novel cellular substance, a phospholipid that he called protagon. However, protagon turned out to be little more than just contamination with the phospholipid lecithin. And as you can imagine, that dented the reputation of Hopper Zeiler's lab somewhat. It cast a big cloud over novel claims to have found new substances coming out of Hopper Zeiler's lab. And this is the environment into which Miescher is announcing his discovery. And not only that, but Hopper Zeiler has a few long-standing feuds with other eminent figures in this emerging field of physiological chemistry. So Miescher makes his announcement of his new discovery against this backdrop of suspicion about novel claims coming out of Hopper Zeiler's lab, and also against this backdrop of feuding, which might account for the reception of Miescher's announcement. So one particular antagonist of Hopper Zeiler was this guy Ludwig Thudichum. He himself was German. He'd fled the German Confederation in 1848 after the revolutions there as a political emigre, because politically he was quite liberal. So he came to London, and he began a meteoric rise through the British medical establishment. Ended up writing in the chief medical officer's reports to the Privy Council, and he was very suspicious and dubious about Miescher's claim that nuclein was a novel material. Other detractors, the clinicians Charles Kingset, William Hake, who described Miescher's nuclein as the great unwashed, so the insinuation being there that once again, like Liebreich's claim on protagon, nuclein is nothing more than just an artifact arising from contamination. So this is interesting, because the biggest source of resistance to Miescher's announcement is coming from within this field of physiological chemistry. What I find ironic is that support for Miescher's claim came from the very discipline that Miescher himself was critical about. That is, the histologists, the cytologists, who do their work by looking at cells down the microscope, cells that have been stained with dyes. A discipline that Miescher himself had once dismissed as being the guild of dyers. You had Edward Zacharias here on the left, who he showed that nuclein, chromosomes were made of chromatin. Oskar Hertwig showed that chromosomes contain nuclein, and from their observations of chromosome behavior, guys like this were already suggesting that maybe chromosomes are the means by which biological traits are transmitted, and that maybe nuclein is absolutely central to that. So I find the irony of this absolutely superb. Oskar Hertwig said, "I believe I've made it at least highly probable that nuclein is the substance that is responsible not only for fertilization, but also for the transmission of hereditary characteristics." So that very field of which Miescher was so dismissive was actually his biggest source of support. I mean, talk about biting the hand that feeds you. Now, what is Miescher doing while all this feuding and fighting is going on over nuclein? Well, by this time he's returned from Tübingen. He's gone back to his hometown of Basel, and aged only 28, he's taken up professorship there, a professorship vacated by his uncle Wilhelm Hess. And it's in Basel that he finds, thankfully, he no longer has to extract his nuclein from pus washed out of surgical bandages, because there is a whole new source of nuclein. There's a huge salmon fishing industry in Basel, and the Rhine salmon has this curious metabolic trick. It will swim 500 miles from the North Sea upstream into the Rhine to its spawning grounds. And during that time, for months and months on end in the spawning grounds, the fish won't eat. Instead, they undergo this kind of anatomical, metabolic remodeling by which they break down their muscles, and all the nutrients from the muscles are used to sustain the fish, but also to build up the sex organs. So there's this huge increase in the percentage body mass of the sex organs of the Rhine salmon. And that's because they're stuffed full of nuclein, what we later call DNA. So when Miescher returns to Basel, he starts working with his uncle because he's become interested in fertilization, and the eggs of the Rhine salmon are a really good material to work with. But he starts going down to the banks of the Rhine early in the morning to catch Rhine salmon, and then bring them back to the lab and extract their testes, because their testes are bulging with this stuff, nuclein, that he worked on in Tübingen. Eventually, right? I mean, I think looking in the sources, he went through about 2,000 Rhine salmon later on. Eventually, I think he cottons onto the fact it makes much more sense to go and see a chap, Mr. Glazer's son, who is like the biggest salmon merchant in Basel. So this was, you all know, there's an old Germanic legend that there was sunken treasure, about the treasure of the Rheingold. Miescher's going down to the Rhine in search of hidden treasure, but for him the treasure is the Rhine salmon. If you remember that Germanic legend about the Rheingold, though, you'll remember the Rheingold brings with it a curse. And in a sense, the Rhine salmon, much as they were a great source of nuclein, they also brought a kind of curse. And that's because Miescher became fascinated by this metabolic trick by which the Rhine salmon breaks down its muscle and lives off the nutrients from that. So he began to investigate this process. He began to do all sorts of research into the metabolism of the Rhine salmon and its diet. As a result of that, he became involved in working for the local fishing industries on the diet of the Rhine salmon, and this began to take up a lot of his time. As a result of that, the work on the Rhine salmon, he came to the attention of the Swiss government, and they got him then working on the diet of inmates in Swiss prisons. Now, at the same time, he's moving up the academic career ladder. He's taking on more and more administrative duties. I'm hoping that if any of you have worked for an appreciable time in academia, or probably any large institution, you'll recognise what's going on here for Miescher. It's a degree of mission creep. In other words, it's that steady dissipation and siphoning away of your initial focus so that you end up being spread out way too thin. And you'll all be familiar with the kind of thing. Would you just sit on this committee? Oh, you wouldn't mind being on that steering group, would you? And gradually it dissipates his energy. And he knew it. He knew it. He complained. He said, so many different things are gnawing away at me. It becomes impossible to achieve one's goals. I'm sinking under 12 hours of weekly lectures, all without any assistance. He talked about the work he did on prison diets. He says, I've been bound in chains with the most arduous and thankless task of my life. I've made myself too green, and now the goats are eating me, he says. Inquiry on the diet of Swiss people, a cookbook for the workers, nutrient tables for the national exhibition, controversies with the Charmer Milk Company. In brief, I'm on the best way to becoming the guardian for the stomachs of all 3 million Swiss. At one point he just cried in exasperation, I need 50 hands, not just 2. Year after year I'm busy from early in the morning until midnight, and there'll be few of my colleagues who take any less rest. But the task is like the Lernaean hydra. For every head that's struck off, 6 more grow in its place. Now, he loved his allusions to classical mythology. I think the most moving one for me was I found a letter where he basically compared himself to Sisyphus in Greek mythology, the poor guy who's doomed by the gods of Olympus to spend eternity rolling a boulder up a mountain. So while Miescher's struggling with his Sisyphean boulder, a guy called Albrecht Kossel, who'd worked with him for a time, chemically analysing Miescher's nuclein and showing that it's made up of these four chemicals: adenine, guanine, cytosine, thymine. He gives them those names. So those are the famous A, C's, T's and G's that you might see when you see DNA sequences today. And the other big development comes from one of Miescher's former students, this guy on the right, Richard Altman, who now rebrands nuclein, gives it a new name, nucleic acid, and the name sticks. And Miescher's not really happy about that, because Altman is trying to claim that Miescher's nuclein was actually impure. It was a mixture of nucleic acid and protein, whereas Altman is saying, but I have actually isolated the pure stuff, the nucleic acid. And I think Miescher felt like he was being a little bit usurped by his former student there. But he's got other things to worry about by then, as Miescher, because he's developed this dry cough. He's getting steadily weaker. The work is taking his toll on him. He's subjected himself to a grueling working regime. And most of all those years of working under freezing cold conditions to stop his nuclein from degrading are taking their toll, and he's diagnosed with tuberculosis. So he starts visiting a sanatorium in Davos, and eventually he becomes a permanent resident there. In 1895, aged only 51, he dies of tuberculosis. And he dies, as I said at the start, he dies with a burdened by a great sense, I think, of failure, of never having lived up to expectation, of opportunities missed. There's a bit where he says, "Only when, here and there, I read in the published work of another author a half-formed fragment of that which I have put together, do I realise what could have been achieved with my material." Another occasion he says, "I will never know the happiness that belongs to the man who has lived up to their station in a harmonious way to the satisfaction of themselves and others." And he basically said, "I go to bed each night feeling like a schoolboy who has failed to complete his tasks," which is there's real tragedy and pathos to this guy. And another great lament of his was that chemists and physicists, he felt, hadn't really thrown their energy into physiological chemistry. So he dies. What happens in the wake of his death? Well, by the start of the 20th century, nucleins hit a couple of problems. I said earlier it was the cytologists looking down their microscopes who were offering the greatest support for it, but now there's a bit of a problem, because when they look down their microscopes, they see that the chromosomes seem to appear and disappear over different stages of the cell cycle. Now, today we know that's not the case. The chromosomes are always there. It's just at certain stages of the cell cycle, they're in a conformation that takes up the dyes more readily, that show them staining under the microscope. But that wasn't known. So chromosomes seem to disappear. And if chromosomes are made of nuclein, then nuclein's disappearing as well. And if the nuclein content of the cell is variable, how on earth can it be the carrier of hereditary information? Another problem came from biochemical analysis, thanks to this guy here, Phoebus Aaron Levine. The four bases I mentioned that Albrecht Kossel had shown DNA was made of, Levine seemed to think that DNA was basically just a monotonous repeat of these four bases, and he therefore thought it was just too monotonous a molecule to have enough complexity to carry biological traits. The best way to understand why that is, think of an alphabet. If an alphabet's only got 4 letters in it, how many words can you generate? Compared with the other big macromolecule in cells, proteins, which are made of up to 20 different amino acids. So you're contrasting a chemical alphabet of only 4 letters with a chemical alphabet of up to 20 letters. By that contrast, it seems like proteins are capable of much more variation and therefore more likely to be the carriers of genetic traits. But things began to change in 1944. This was a pivotal year. Firstly, chap on the left here, Oswald Avery, showed that in bacteria at least, nucleic acids could pass on the ability to cause disease. The other big development was a book written by Erwin Schrödinger here, who's probably more famous for his role as a founder of quantum mechanics and the infamous thought experiment with the cat. that's neither dead nor alive, and which has been many a plot device for the hit science sitcom Big Bang Theory. So in 1944, Schrödinger had this, while he was a political emigre in Dublin, Schrödinger had this book published, What is Life? Where as a physicist he tried to think about the phenomenon of life. Now, much has been claimed for this book in the history of molecular biology. A few years ago, Professor Brian Cox, doing his series Wonders of Life on the BBC, talked about the importance of this book in the first series. Many physicists have claimed that this was the call to arms that drew them from physics into biology. A few here, Gunter Stent, Max Delbruck, perhaps more famously, both Maurice Wilkins and Francis Crick said reading this book was really what got them to turn their attentions to biology. I would say that James Watson as well, there is a letter from him to Schrödinger saying, like, dear Professor Schrödinger, I just wanted to say how influential your book's been. And this is why, going back to the plaque in the eagle, this is why I think maybe actually Miescher would have been delighted to see this plaque in the eagle, because the knowledge, if you remember I said, one of his laments before he died was that physicists and chemists were not really showing much interest in physiological chemistry. To see that plaque and to know that of the 3 Nobel Prize winners for the double helical structure of DNA, 2 of them were physicists who turned their attentions to the problem of heredity. I think Miescher might have found some consolation with that. Just as a little aside, I should say I don't completely buy the argument that What is Life was the rallying cry to get physicists into biology, because about 6 years before What is Life was published, 2 physicists, William Astbury and Florence Bell, were actually applying experimental techniques to the study of DNA, and they'd done that without reading What is Life. Not everyone was impressed with What is Life. Linus Pauling said, I was disappointed, and Max Perutz said, well, what was true in the book wasn't original, and most of what was original wasn't true, which I always thought was a little bit harsh, because I always thought something that Schrödinger did say that was original was he identified the 2 key properties a genetic molecule must have. It's got to be big, and it's got to be capable of variation. How could it be capable of variation? Well, Schrödinger said one way it could do it would be through stereoisomerism. So stereoisomerism is where we have chemical with the same elements, the same chemical structure, but they're arranged differently in space, in the same way as left and right-handed gloves. In both cases, you've got 4 fingers and a thumb, but there's no way you can superimpose those on each other in space. And I always thought that was Schrödinger's big idea, until I discovered Friedrich Miescher. Because alongside his discovery of nuclein, I think Miescher made another really important contribution. And it's a letter that he wrote in 1892, about 3 years before he died. And he said, to me, the key to the problem of sexual reproduction is to be found in the field of stereochemistry. He said, the huge molecules of albumin compounds, by which he means proteins, the many asymmetric carbon atoms provide a colossal amount of stereoisomerism, and in them all the wealth and variety of hereditary transmissions can find expression, just as all the words and concepts of languages can find expression in 24 to 30 alphabetic letters. So, again, so actually what Schrödinger was saying there is not that novel, because Miescher had said it. Now, as a historian of science, I've got to say here, it is not the job of historians of science just to pass judgment on who got there first, who got over the finish line, as if we were some kind of historiographic VAR device in a football match, judging whether the ball was over the line. Miescher's letter has been described as an early reference to genetic coding. I think we're getting a bit too excited there. The modern concepts we have as DNA working as a code were not part of the intellectual landscape of Miescher. Both Lily Kay and Matthew Cobb have shown in these 2 books that those ideas of coding came into molecular biology in the 1940s through the mathematical branch of information theory and cybernetics. But nevertheless, I think we've really got to hand something to Miescher for this. Why does his story matter today? Well, I mean, there's not a week goes by, you won't flick open the newspapers, you'll see some talk of DNA. The legacy, the technological legacy that has come from his discovery 3 simple examples: production of drugs using recombinant DNA, such as insulin, CRISPR gene editing that we're hearing more about, mRNA vaccines, for which we were also thankful during the COVID pandemic. But I think, to finish, there's another reason why his story matters today. So this is my late colleague, John Hodge, who sadly passed away last month. John was a great source of support when I moved from science into history of science. But the most important lesson I learnt from John and was in this lecture he gave here in 2016 as part of our History and Philosophy of Science in 20 Objects lectures. John is making a plea in this picture, and his plea is this: for us not to be seduced by the allure of overly simplistic, reductive narratives. Now, John was trying to cultivate in burgeoning history of science students a critical approach to historical narratives, but actually, as I look around the world in 2025, I think that John's warning about being seduced by simple narratives rings true beyond the seminar rooms and the lecture theatres of academia. You will know the kind of thing that John is warning against. History of science is full of these oversimplified narratives. We love tales of lone geniuses who are somehow ahead of their time and eureka moments. Archimedes in his bathtub, Newton under the tree. Those stories they're misleading about how science really evolves. They obscure how science really develops. And worse than misleading, I'm going to finish with one short example that such simplistic narratives are actually dangerous. So you may remember of 2018, scientist here, Professor He-Zhong Cui at the University in Shenzhen, got up at this second International Summit on Human Genome Editing and announced that using CRISPR gene editing technology, he had made deliberate alterations in the DNA of 3 human embryos that had then gone fully to term and been born as 3 little girls. Now, Matthew Cobb covers this story really well in this book, The Genetic Age. He-Zhong Cui's announcement was met with outrage and condemnation. I won't go into the details of what he did. It's a talk in itself. He basically used CRISPR gene editing to make edits in a gene that was thought to be involved in reducing susceptibility to HIV infection. When put on the spot and asked what was the immediate life-threatening need that justified you making that intervention, he couldn't give an answer. But he later said he was doing it to try and make the girls immune to HIV. Well, as Matthew Cobb points out in his book, if you don't want to catch HIV, there are far more simple methods of avoiding HIV than having to get your genome altered using CRISPR gene editing. As a historian of science, what I find interesting about this episode is that He-Zhong Cui was seduced by 2 overly simplistic narratives, one of which is bad science. So here are a few sample headlines. You will find headlines like this, I think, most weeks in any newspaper, and it's this idea, beloved of the media, that there is a gene for any human trait, behavioural, physiological, there is a single gene for it. As if genes are just a row of buttons and you press one and you get blue eyes, and you press another one and you can play the violin, and you press another one and you can run fast. I hope it comes as no surprise to you to say genes are way more complex than that. Single genes do multiple functions. They do different things depending on what other genes are switched on around them, what cell types they're in, what stage of development the organism is at, and crucially, what environmental influences they're subjected to. So this notion of there simply being a gene for is ridiculously and dangerously deceptive and oversimplified. The second seductive influence, overly seductive, simplistic narrative that He-Zhong Cui fell for was bad history, or what I've called cowboy science. As a young scientist, He-Zhong Cui was told by a senior scientist that science progresses through people who are not afraid to break the glass, or we might say today, move fast and break things. It's done by people who do cowboy science, you know, the kind of thing, the maverick, the person who's not afraid to play a bit fast and loose with the rules. So He-Zhong Cui went away and he immersed himself in that kind of historical narrative. And the one in particular he immersed himself in was Edward Jenner. And he thought, I want to be that kind of guy. Jenner wasn't afraid to break the rules a bit and do cowboy science. That's who I want to be. Well, again, probably comes as no surprise, but the story of Jenner and the smallpox vaccination is much more complex than that. But He-Zhong Cui was seduced by that, and it resulted in him getting a 2-year prison sentence. Just to finish, I would suggest that we would be better off, rather than trotting out these oversimplified narratives of scientific heroes, a far better image for the advance of science is Miescher as Sisyphus struggling up that mountain, and the rock gets passed on. It's a team effort, and the rock gets passed on from one group to another. So I'm going to finish there. I just want to say thank you in particular to Dr. Neeraj Sankaran, who first brought Ralf and I together in a podcast on Miescher, and also to Dr. Frida Trotter, our editor at Springer Nature, who's been so supportive and patient with us throughout this. And now I'd like to hand over to Ralf to talk about his role in really making sure that Miescher is remembered, and also, and I'll say my thanks to you all for listening so patiently. So Ralf, if you would like to take over from there.
Good. So thanks for passing on the boulder, Kersten. I will continue with a bit of a personal story, much briefer than what Kersten laid out. And it will not be so much about the reception of Miescher's work in the scientific community. That has been covered by Kersten. I will talk about the reception of Miescher's discovery in the city where he made the discovery, Tübingen. This is an old picture taken round about the time when Miescher made his discovery of Tübingen Castle. Sounds fine, I'm just reading, cool. And I will just switch on my laser pointer. So this is the castle perched high on top of a hill above the old town of Tübingen, where Miescher worked from 1968, 1969 to make his discovery of DNA. This is, as Kersten showed, Miescher's laboratory, where he made his discovery with very basic equipment compared to what we find in modern laboratories today. And the question for me when I moved to Tübingen in the late 1990s was, how does the city remember one of the greatest discoveries made in the life sciences? And this is the answer. What you see here on the left, panel A, is a picture of the inside courtyard of Tübingen Castle. And right next to this door, which actually leads to the former kitchen, to the laboratory, this one, where Miescher made his discovery in. Right next to this door, there is 3 plaques which commemorate important events in the city's biochemical research. Most importantly, Felix Hoppe-Seyler has a big plaque on the occasion of his 100th birthday. Hoppe-Seyler, as Kersten mentioned, was one of the pioneers of the then very new discipline of physiological chemistry, or biochemistry, as we call it today. Made seminal work on hemoglobins and many other aspects, and definitely was an important biochemist, although his discoveries arguably don't quite compare to Miescher's. There is a second plaque on something unrelated, and then there is a plaque actually mentioning Friedrich Miescher. But what it says is that on this spot, Friedrich Miescher in 1869 discovered nuclein. And when I came to Tübingen in 1999, I had no idea what nuclein was. I didn't even remember the story of Miescher discovering DNA anymore, although I'm sure I've read it as a side remark in some textbook on the history of molecular genetics or whatever it may have been. I was lucky, however, in that I had a supervisor at the Max Planck Institute where I was working at the time, who was very historical minded. And she kept pointing out to us that we lived in a very historic context, and that, amongst other things, DNA had been discovered in Tübingen. So I was primed. And then in 2003, the 50th anniversary of Watson and Crick's discovery of the double helical structure of DNA, which Kersten also mentioned a minute ago, had its 50th anniversary. And on this occasion, all the newspapers, the magazines, the scientific journals were full of articles, editorials, news and views pieces commemorating this monumentous event. And no one ever mentioned Tübingen. No one ever mentioned Friedrich Miescher, not even in the city where he had discovered DNA. I thought that was a bit of a shame, so I set out to write an article for the local newspaper, the Schwäbisches Tagblatt. Don't worry if you've never heard of it. It's a very provincial newspaper, which is published only in the small town of Tübingen. But I felt at least the people living in the place where DNA was discovered should know about it. And things, for me, that was the end of it. I was prepared to let go and, have others have their say in this. But it wasn't to be, because this article was seen by the editor of the Max Planck journal at the time. And he asked me if I couldn't write a piece on Miescher's discovery of DNA for Max Planck Research, which is their Max Planck Society internal journal. So I did. And again, I thought I'll leave it at that. But no, our institute was visited by the editor of Developmental Biology, another, this time scientific journal in the biological sciences. And he saw the article when he was staying in our guest house. The journal was lying there. He saw the article on Miescher, which I'd written for Max Planck Research, and asked me if I couldn't contribute to his journal a piece on the discovery of DNA. So I did. And to cut a long story short, there followed many other articles, for example, for Human Genetics, for EMBO Reports, for bioessays, and also more in the popular scientific journals, such as American Scientist and The Scientist. So ultimately, I'd written a long range of articles on the history of DNA without even coming close to being a historian. And I was at the time even asked to write a book by Springer, the German publishing group, asked to write a book on the discovery of DNA. And I had sort of grudgingly agreed to do it with a colleague because I felt I wasn't really the right person to do it. And then I was incredibly fortunate in being introduced to Kersten in the podcast he just mentioned. And he was the perfect person to write that book because he actually is a historian of science and knows what he's talking about, very much in contrast to me. And this is the result, which will be published, as Kersten mentioned, soon, a book which personally I can say I think it's a fantastic book. If you have any interest in the history of DNA, the early research into DNA, how it was received early on, all the way up to now, how our DNA matters so much for us as scientists, but also as a society at large. This is really a very good book to read. And I'm completely unashamed to sing its praises because this book is so fantastic, not because of me, although my name appears in the same font size as Kersten's, but it really is down to Kersten, who has put an extraordinary amount of effort into researching all the side angles of the early research into DNA and whatnot. So it truly is a very insightful read. And my name should be printed in a much smaller font size, I should say, than Kersten's, because it really is his achievement here. So if you're interested, go buy the book. I think you will not regret it. It will be a very enjoyable read. Also very accessibly written, I find it. So Miescher, since then, like I said, went to Tübingen in 1999. Miescher was pretty much ignored. And I was fortunate in another sense as well, in that I met the founder and CSO of the company CureVac, which is likely a company you almost heard of. I say almost because, like BioNTech and Moderna, they were very much working on the same vaccines for COVID, only they failed very narrowly, unfortunately. But if they had done slightly different chemistry, they now would be as famous as BioNTech and Moderna are for their RNA-based vaccines. But they did make enough money. And Ingmar, the founder of the company, was sufficiently enthused about the history of DNA's discovery and things that they donated a substantial amount of money to the University of Tübingen to found a museum in the castle, in the laboratory, in fact, where Miescher worked and where Hoppe-Seyler worked. So instead of this being a storage room for archival material of the archaeology department, which it was when I was there for the first time, it now is a museum dedicated to the history of DNA's discovery. I'm just flicking through a few pictures so you can get a little bit of an impression of what the museum now looks like. And I can only say it's a nice mix of multimedia displays and historic artifacts, historic samples, equipment and things. If you ever get a chance to go to Tübingen, I would advise you to visit the place. Tübingen Castle in and of itself is very much worth a visit, different museums in there, including for archaeology and antique sculptures and things. But now also a little museum on the history of DNA's discovery. And I'm glad to say that nowadays there is even a trilingual plaque outside Tübingen Castle where which refers to the discovery of DNA, not only nuclein, by Friedrich Miescher there in 1869. And with this, I would like to finish. I'll leave you with a contemporary picture of what Tübingen Castle looks like nowadays. And I guess both Kersten and I are very happy to take any questions, should there be any. Thank you very much.
Yeah, now, Ralf, I've just got to say that, hey, listen, the book was thank you for your kind words, but the book was a team effort. Yeah, just going back to Miescher rolling that rock, that was the two of us, me and you, rolling that rock up the mountain. So there you go, double Sisyphus.
Thank you very much. Thank you very much, both of you, for a wonderful talk and a wonderful background explanation of how the book came to be. I must say, if I may make one remark first, a supervisor of mine when I was at Oxford University wanted to send me to Tübingen for a year. And I don't quite know why it didn't happen, but seeing these pictures, I very much regret that it didn't happen. So he did say it was a very beautiful place, but for some reason it never happened. And by the way, plug for my book. My book, The Polymer Revolution, is coming out later this week. It's been published by the RSC. Please all rush out and buy it. That's my little plug ended. Now, you've had your plug. I've mentioned to people, by the way, there's a sale at Springer today, just for today, where you can buy a Springer book for £18. So I don't know if your book is part of that sale, but I'm going to look and see if we get off this talk. Now, my question to you is that it's a very interesting person I looked into when I was at the Science Museum, because I had a chemical, or 150-year-old chemical, and his name might be familiar to you. It's Ludwig Friedrichsson. Have you come across Friedrichsson? No, you're muted. You're muted.
Yeah. So Friedrichsson, he's on my slides.
Okay.
We really dug into his story. There's loads about him in the book because
particularly by a chap called Drabkin.
I've read that. Yeah, no, Drabkin calls Friedrichsson the first British biochemist.
Yeah, that's that. Now the point is, but he got into a dispute, at least with Hopper Saylor, if not other people as well, about the nature of nuclein.
Yeah, but the thing is that the punch-up between Hopper-Seyler and Friedrichsson was already they were already crossing swords before nuclein came along, because I think they'd had a disagreement about hemoglobin. They had so basically it's like but
the big argument was about a chemical that doesn't really exist because it's a mixture of chemicals called protogon. He isolated a substance from the brain called protogon.
That's right. It was Liebreich in Hopper-Seyler's lab. Yeah. And I kind of picture Friedrichsson and Hopper-Seyler, Friedrichsson in the blue corner of the boxing ring, Hopper-Seyler in the red corner, and then poor old Miescher gets up into the middle and makes his announcement as the two of them are just trading blows. And it makes for interesting reading, actually, reading their exchanges in the pages of the journals, and the way they snipe at each other, undermining, and I suppose we'd call it gaslighting today, on the journals, all the various insinuations, and it gets quite nasty, and made for very interesting reading.
Well, it's also interesting because Friedrichsson himself has been very much overlooked. In fact, paradoxically, if you ever come across Friedrichsson today, it's more for his nasal spatula. He developed a tool for opening up the nasal passage. He was an ENT, among other things. He was an ENT surgeon. And he invented this sort of spatula to opening up the nose. And that's what, if you ask an ENT person today, he will know about the Friedrichsson tool. Because
it sounds like he had quite an eclectic research program, Peter, because I found one publication where he was talking about an experiment into alcohol intake that he'd conducted using students. And he basically just saying how many bottles of wine everyone had quaffed. And there's also fascinating, he has this...
he came from a wine area. I can't remember exactly where. It was near Frankfurt, I think, but he came from a wine growing area.
Ah, right,
So
yeah.
he's also a great expert on wine. So he was a great expert on nosy. He was a great expert on wine, and he considered himself to be a great expert on the
And
burn.
also added to that on the health benefits of the Turkish bath,
Yeah.
which the
Yeah, that's a new one. I haven't heard that one.
oh no, there's an ongoing exchange on the pages of the Lancet and the BMJ.
Yeah, right.
Oh yeah, the Turkish bath was
They do
controversial.
think the turkey bath. They do think the turkey bath is good for you now because it opens up the pores and all sorts of
Yeah.
other stuff. Anyway, coming back to just the Buddhism for a moment, of course, it's fascinating about Buddhism, and this does have some bearing on Miescher, is that he studied the brain because there was so much phosphorus in it. So that goes back to your comment at the very beginning about phosphorus. But the other reason why it's been so overlooked is because he assumed that it was the big, it was the chemicals that you found in the actual structure of the brain that mattered. Because he was convinced there was a connection between the brain and chemicals. But he got the wrong chemical. He looked for all the big chemicals, and what he missed was the fact that you need to look at the very tiny neurotransmitters and other chemicals in the brain that really matter. So nowadays, people are interested in brain chemistry, but it's a completely different type of chemical. But anyway, that's good. Now, I think that is all from. Did I have another question? I think that might be about it. But we do have one question from the audience. Oh, by the way, could I say just before I move to the question, which is a very good question, greetings everybody from the Leeds Literary and Philosophical Society, who have all been brought here by Christine Hostock. If any of you are interested in seeing any more of these talks, please get in touch with me via Christine and I'll add you to my personal mailing list. Sorry again for that other plug. Now, Sister Bat asked the question, was there any experimental evidence provided by Hertwig on the presence of nuclein in chromosomes?
Yeah, now I've messaged right, so I messaged Shrisha back. Yes, I checked it out. So Hertwig had gone from Berlin to the French Riviera to work on to observe sexual reproduction in sea urchins. As you,
right.
He was probably glad of the sunshine down there. And he did. So he found that the two sets of chromosomes come together. But he did staining experiments which showed that chromatin, the material from which chromosomes were made, was predominantly nuclein. And so from that, he starts to make the inference that, right, nuclein must be absolutely central in this reproductive process. So I dropped Shrisha a message back just to say that.
Okay. Well, I think that's very good of you. You're the first speaker who's ever done that. So thank you for taking care of our visitors. Now, may I just say to everybody, well, once again, first of all, to our speaker, Kersten Hall, another fantastic talk by Kersten Hall, and thanks, Ralf Dahm, for bringing in the German angle. Fascinating place, Tübingen. I must go there sometime. And thank you, everybody, including, of course, especially the people from the Leeds Literary and Philosophical Society, for coming along to the talk. And I hope that you consider coming to one of our talks in the future. Next month, third Tuesday of the month, that's when we always meet. 2 p.m., that's always the time, same back channel, as they say. And it's going to be about recreational chemistry because it's Christmas. And it's going to be given by Colin Johnson, who was the director of Techniquest in Cardiff. So I'm sure that'll be a fascinating talk. He's been working on it for a long time, I know. And please come along and have some Christmas fun with us next month, 16th of December, if I remember rightly, but it's the third Tuesday. And these talks will continue at least for a few more months, but I'm going to step down in March. However, we're going to have a meeting next week to see how we can continue. So once again, thank you very much, everybody, and have a nice afternoon. Bye for now.
Thank you, everyone. Thank you.